Welcome to Frontiers of Research. I'm Antoni, and today we're exploring the revolutionary field of spatial biology and cell atlas projects, efforts to map the human body at unprecedented cellular resolution while preserving crucial information about where cells are located and how they interact with their neighbors. Joining me are Sarah, who works on single-cell genomics technologies, and Josh, who's been following the historical development of cell mapping efforts. Together, we'll discuss how these new approaches are transforming our understanding of human biology.
Thanks, Antoni. To understand why spatial biology represents such a breakthrough, it's helpful to look at the limitations of earlier approaches. For decades, biologists have studied tissues by homogenizing them, essentially blending them up to extract biomolecules like RNA or proteins. While this gave us valuable information about what genes were expressed or what proteins were present, we completely lost information about which specific cells these molecules came from and how these cells were arranged. Then came single-cell technologies that let us analyze individual cells, which was a huge step forward, but cells were still removed from their native context. Spatial biology combines the resolution of single-cell approaches with spatial context, allowing us to see exactly which genes are active in which cells while preserving information about where these cells are located within tissues. It's like the difference between having a list of all the parts in a car versus having a detailed blueprint showing how they fit together.
This field actually has fascinating historical roots. In the 19th century, anatomists like Santiago Ramon y Cajal were painstakingly mapping the locations and connections of cells using microscopy and histological staining. Cajal's intricate drawings of neurons are still used in textbooks today. Then in the mid-20th century, techniques like in situ hybridization were developed to visualize specific RNA molecules within cells. But what's happening now represents a genuine revolution. The convergence of several technologies that collectively allow us to map thousands of genes or proteins simultaneously across entire tissue sections, with cellular or even subcellular resolution. It's a bit like the difference between early explorers mapping coastlines versus modern satellite imaging that can resolve individual buildings. What's particularly exciting is the scale and coordination of today's efforts compared to the relatively isolated work of individual labs in the past.
Sarah, could you walk us through some of the key technologies that have enabled this spatial biology revolution?
There's been an explosion of methods in the last few years, which I'll broadly group into a few categories. First are spatial transcriptomics platforms that measure RNA. These include methods like Visium from 10x Genomics, which uses arrays of barcoded spots on slides to capture RNA from tissue sections. Each spot has a unique barcode, so when RNA is sequenced, we know which location it came from. More recently, technologies like SlideSeq and high-definition spatial transcriptomics have pushed resolution down to individual cells. Then there are in situ sequencing methods like MERFISH and SecuFISH Plus, which directly visualize multiple RNA molecules within intact tissue using fluorescence microscopy and sophisticated barcoding schemes, allowing simultaneous imaging of thousands of RNA species. For proteins, there are multiplexed imaging technologies like Codex and Vectra that use cycles of staining, imaging, and signal removal to visualize dozens or even hundreds of proteins in the same tissue section. The newest frontier is spatial multiomics, where multiple types of molecules, RNA, protein, lipids, metabolites, are measured simultaneously in the same tissue section, giving us a more complete picture of cell state and function.
Josh, can you tell us about the Human Cell Atlas and other large-scale mapping initiatives?
The Human Cell Atlas is an international collaborative effort launched in 2016 with the ambitious goal of creating comprehensive reference maps of all human cells as a basis for understanding health and diagnosing and treating disease. It's often compared to the Human Genome Project in terms of scope and potential impact. But instead of mapping genes, it's mapping cells. the fundamental units of life. This involves cataloging all cell types, their molecular characteristics, and importantly, their spatial relationships. It's a massive undertaking involving thousands of scientists across more than 70 countries. Beyond the Human Cell Atlas, there are other significant initiatives like the NIH's Human Biomolecular Atlas program, HuBBY-MAP, which focuses specifically on spatial mapping of human tissues. The European Lifetime Initiative, which aims to track how cells change over time in health and disease, and the Allen Brain Atlas, which has been mapping mouse and human brain cells for years. What's notable is the open collaborative nature of these efforts. The data and tools are being made freely available to researchers worldwide, accelerating discovery in ways that wouldn't be possible with more siloed approaches.
Sarah, these technologies must generate enormous amounts of data. What computational tools and approaches are being developed to analyze this data?
That's absolutely right, Antony. The computational challenges here are immense and have spurred the development of entirely new analytical approaches. A single spatial transcriptomics dataset can contain information about thousands of genes across tens of thousands of locations, creating matrices with millions of data points. Add in the third dimension for 3D reconstructions, or the fourth dimension of time for developmental studies, and the complexity grows exponentially. Specialized tools have emerged for different aspects of analysis, from pre-processing and quality control to segmentation of cells or tissue regions, and spatial statistics to identify patterns and interactions. What's particularly exciting is the application of deep learning approaches to extract patterns from these complex datasets. For example, convolutional neural networks originally developed for image analysis are being adapted to identify spatial patterns of gene expression. There are also methods for integrating spatial data with other data types like single-cell RNA sequencing or chromatin accessibility data. One fascinating area is the development of algorithms that can infer cell-cell communication networks based on the expression of receptor-ligand pairs in neighboring cells, allowing us to decode the language cells use to talk to each other.
Let's talk about how these technologies are being applied. What are some of the insights or applications that have emerged from spatial biology approaches?
The applications are extraordinarily diverse. In cancer research, spatial biology has revealed the complex ecosystems of tumors, showing how cancer cells interact with immune cells and how these interactions influence therapy response. For example, researchers have identified specific spatial arrangements of immune cells in tumors that predict whether a patient will respond to immunotherapy. In neuroscience, these approaches have uncovered previously unknown cell types in the brain and mapped the circuits they form. In developmental biology, spatial transcriptomics is revealing how patterns of gene expression drive the formation of complex organs. There's also growing clinical application. Spatial pathology platforms are being developed that could transform diagnostic procedures by providing more detailed molecular information than traditional histopathology. Perhaps most fundamentally, spatial biology is challenging some of our basic concepts about cell types. We're finding that the identity and function of cells can be heavily influenced by their location and neighbors. The same type of cell might behave quite differently depending on its spatial context. This is forcing us to reconsider how we classify cells and understand their roles in health and disease.
What are the current limitations or challenges in the field of spatial biology?
Despite the remarkable progress, significant challenges remain. There's still a trade-off between the number of genes or proteins that can be analyzed and spatial resolution. Methods with the highest resolution typically measure fewer molecules, while those that measure more molecules often have lower resolution. There are also technical challenges with tissue preparation and artifacts introduced during processing. Data analysis remains complex, requiring specialized expertise that isn't widely available. And generating these datasets is still expensive and time-consuming, limiting the number and size of studies. Another major challenge is integrating data across different spatial scales, from subcellular structures to cells to tissue regions to whole organs. Ideally, we want to zoom seamlessly across these scales, but that's technically very challenging. Finally, there's the challenge of moving from descriptive maps to mechanistic understanding. Knowing where cells are and what genes they express is just the first step toward understanding how they function together as complex systems.
Looking ahead, what do you both see as the most promising future directions for spatial biology?
I think we'll see a continued expansion of these efforts to map more tissues, organs, and even entire organisms at different developmental stages and in various disease states. The Human Cell Atlas and similar projects are really just getting started in terms of the breadth of tissues and conditions they're examining. I also expect we'll see these approaches applied to more diverse human populations to capture biological variation across humanity. And from a historical perspective, it's fascinating to consider how these incredibly detailed molecular maps might change our fundamental understanding of biology, much as the first anatomical atlases revolutionized medicine centuries ago. The ability to see the molecular composition of tissues in their native context could lead to entirely new biological principles that we can't even conceive of yet.
Building on what Josh said, I think technology development will continue at a rapid pace. We'll likely see methods that can capture more molecules at higher resolution, eventually approaching the goal of measuring all RNA and proteins in every cell while preserving their spatial context. There will be more emphasis on dynamic processes, not just static snapshots, but how spatial patterns change over time during development or disease progression. I expect greater integration of spatial methods with functional approaches that perturb cells and observe the effects, which is essential for moving from correlation to causation. The computational methods will continue to evolve as well, with more sophisticated approaches for integrating diverse data types and extracting biological insights. Finally, I think we'll see increasing clinical translation, with spatial pathology becoming standard for cancer diagnosis and potentially for other diseases as well. This could fundamentally change how diseases are classified and treated, moving us toward truly personalized medicine based on the unique cellular and molecular architecture of each patient's tissues.
Thank you, Sarah and Josh, for this fascinating exploration of spatial biology and cell atlas projects. We've covered how these approaches are providing unprecedented views of the cellular architecture of tissues, the technologies driving this revolution, and the insights emerging from these detailed maps. This field exemplifies the power of combining technological innovation with collaborative scientific initiatives to tackle fundamental questions in biology. As more detailed and comprehensive cell atlases are generated and integrated with other biological data, we can expect transformative insights into how cells work together in health and disease. I'm particularly struck by how these approaches are revealing not just what cells are present in tissues, but how they communicate and influence each other, turning static maps into dynamic blueprints of biological systems. Join us next time as we continue to explore the frontiers of research.